Welcome to Science Makes Me Smile Biomedical. In a new world where engineers, computer scientists, not necessarily medical researchers, are at the forefront of medical inventions, medicine is becoming a new entity to explore, understand, and benefit from. I'm young Dr. JGC, guiding you through in this new adventure. Let's begin. In our first Science Makes Me Smile Biomedical, we're looking at how biotechnology is turning sickle cell anemia from a lifelong condition with no realistic cure into something that, for many patients, can now be treated with curative intent. Sickle cell anemia is an inherited blood disorder where red blood cells can become rigid and crescent-shaped instead of soft and flexible. In the U.S., it affects mostly Black African American people, the large majority of cases, though it can also occur in people with ancestry from other malaria region populations. What makes it so serious and so painful is the traffic jam effect. Sickled cells don't slide through tiny blood vessels, they can block them. That cuts off oxygen to tissues, triggers inflammation, and leads to the obstruction of the vessels, causing vaso-occlusive crises, episodes of severe pain that can also contribute to complications over time. Most care today is therapeutics, not a cure. That includes pain control during crises, hydration, transfusions when needed, and medicines that reduce crises and complications for many patients. Now here's the wild part. The root cause is a tiny genetic typo with huge consequences. In the HBB gene, a single letter change alters the hemoglobin recipe so that at position 6, glutamic acid, water-loving, charged, is replaced by valine, water-shy, hydrophobic, the classic Glu-6-Val change. That swap changes the surface behavior of the hemoglobin protein. When oxygen is low, hemoglobin molecules start sticking together and form long fibers. Think little sticks inside the red cell, making the cell stiff and sickle-shaped. And it makes you want to ask, wouldn't it be nice to just snap that one little mistake out? But DNA is a library of billions of letters. Finding one typo in the right cells and fixing it without messing up the rest is like doing one-letter surgery in an encyclopedia while the book is still being read. Enter CRISPR-Cas9. Think of CRISPR as the address label and Cas9 as the molecular scissors. A guide RNA brings Cas9 to a specific DNA sequence. Cas9 makes a precise cut, and the cell's repair machinery handles what happens next. Need a repeat? Think of CRISPR as the address label and Cas9 as the molecular scissors. A guide RNA produced by a lab brings Cas9 to a specific DNA sequence. Cas9 makes a precise cut, and the cell's repair machinery takes it from there. With the FDA-approved CRISPR therapy Casgevy, doctors collect a patient's own blood-forming stem cells, edit them outside the body to boost fetal hemoglobin, which doesn't sickle, then give the edited cells back as a one-time infusion after conditioning treatment. And the results so far are striking. In a major report, 97% of evaluable patients, 29 out of 30, had no vaso-occlusive crises for 12 months or more. To appreciate that, remember what before looked like. These weren't mild cases. The baseline rate was around 3 to 4 crises per year, median 3.5 per year, with some people much higher.
Every conversation leaves a trace, not of certainty, but of insight. What you do with it is up to you. I'm young Dr. JGC. Until next time. Comments, questions, suggestions are welcome. Please remember to like and subscribe. Science makes me smile.
In our next episode of Science Makes Me Smile Biomedical, we'll discuss why sickle cell disease clusters in regions where malaria is or was common, and how modern biomedical technology, from vaccines to gene-based tools, is helping push the world closer to malaria control and, ultimately, eradication.
Contact sciencemakesmesmile@yahoo.com.